Yes, good afternoon everyone, and we have completed the biomolecules up to the vitamins, and the next topic we are going to have is nucleic acids. I think everyone knows about where the nucleic acids are present, what is their significance. No need to explain for you, the biology students. The nucleic acids are going to be present within the nucleus, and these are going to be responsible for so many important functions. The main one is heredity and the protein synthesis. These are the main we are going to have for a nucleic acid. How they are going to have the structure, the chemical structure of nucleic acid is going to be studied in our discussion. Now these nucleic acids are the acidic property containing compounds which are present in the nucleus. They are called as nucleic acids. So these nucleic acids are mainly divided into DNA, RNA. So what we are having? Two types of nucleic acids. They are DNA and RNA. DNA full form is deoxyribonucleic acid. Deoxyribonucleic acid, whereas RNA is ribonucleic acid. The DNA full form is deoxyribonucleic acid, and this one is going to be ribonucleic acid. Now the name itself is going to say deoxy, and this one is ribo. Deoxyribonucleic acid, ribonucleic acid. The main sugar moiety present in each nucleic acid is going to chemically differentiating them into two. Already I discussed what sugar is going to be present here. Beta D ribose, where in case of RNA, 2' deoxy beta D ribose in case of DNA. That's what already we discussed. And again we are going to see the structure and all after we are going to discuss something regarding the DNA and RNA. Structural peculiarities we will see later. This is called deoxyribonucleic acid. This is called ribonucleic acid, and this is going to be double stranded. Mostly this is single stranded. Mostly we are going to have some double stranded RNAs, but mostly it is going to be single stranded. This is going to be double stranded. That's what you are going to have DNA and RNA when you are taking. And what ribose sugar we are going to have here, that is main thing I said, it is going to have 2' deoxy beta D ribose. 2' deoxy beta D ribose is going to be present in DNA, whereas here it is going to be present beta D ribose. And if you are coming to this one, the nitrogen bases present are nitrogen bases we are having A, T, G, C. These four nitrogen bases, adenine, thymine, guanine, cytosine will be present here, whereas here we are going to have nitrogen bases are A, U, G, C. Adenine, uracil, guanine, cytosine. So instead of thymine, we are going to have uracil here. That's the difference we are having between the nitrogen bases present in DNA and RNA. Only thymine is replaced by uracil. Understand? And then here we are going to have autocatalytic activity. What activity it is going to have? Autocatalytic as well as heterocatalytic. What is the meaning of autocatalytic, heterocatalytic? Autocatalytic activity is nothing but the DNA can able to synthesize the synthesis of its own. It can able to direct the synthesis of its own, which is called as replication. Heterocatalytic means different. So this is going to be called as transcription. So what we are going to have, the DNA is going to have the synthesis called of RNA, which is called as transcription, whereas the protein synthesis, that is not going to be directed by this one, that is going to be called as translation, that is done by your RNA. So what we are going to have here, autocatalytic function, that is auto means self. It can able to catalyze its own synthesis, that's called autocatalytic function. That is going to be called as a process replication, replica, xeroxing. Replica means taking the copy. DNA can able to take the copy of its own. Whereas hetero means different. Catalytic means catalyzing the different molecule other than the DNA, that is RNA, that's called transcription. Whereas here we are going to have heterocatalytic function only, that is translation. That is called as translation. So translation is going to be the function of your RNA. That is what you are going to have. And the DNA, if you are taking, we can have the different types of DNA based on the number of bases and all. That is aDNA, bDNA. But functionally the DNA is going to have only one function, that is hereditary function. That is hereditary function. Whereas coming to RNA, it is going to have three types: rRNA, tRNA, mRNA. I am going to have three types of RNAs based on their function, but functionality of DNA is going to be one, that is having single function, that is going to be having hereditary function. But in this case we are going to have rRNA, tRNA, mRNA. What is rRNA? It's going to be called as ribosomal RNA, which is present in the ribosomes, and it is responsible for platform for the protein synthesis. It is going to provide platform for protein synthesis. Whereas tRNA is going to be called as transfer RNA, which is responsible for transporting the amino acids from their synthesis to protein synthesis site. Where the amino acids are going to be present, from there it is going to take the transport the amino acids from the site of production to site of protein synthesis. That is what we are going to call as tRNA. mRNA, messenger RNA. This messenger RNA is responsible mainly for the giving the information regarding protein synthesis by its sequence of bases, which is going to be popularly known as genetic code. I think every one of you are aware of that one. Genetic code is going to have 3-letter code. Every 3-letter is going to be responsible for giving one amino acid. So that is going to be taken care by mRNA. So this is what we are going to have, the different types of RNAs and their roles, and this is going to be the DNA. So this is regarding basic differences what we can able to find for DNA and RNA. Now we will see structurally what differences we can have for the DNA and RNA. I hope everyone can understand now this one. DNA is going to be deoxyribonucleic acid, which is going to have deoxyribose. RNA is a ribonucleic acid, which is going to be having ribose sugar, whereas it is double stranded and it is going to be single stranded and it is going to have nitrogen bases ATGC. Here I am having AUGC. Here it is going to have two functions, transcription and replication. It is going to have translation. Functionally, the DNA will not have any differences we are going to have, but structurally we are having aDNA, bDNA, zDNA, all these are going to be, they are having some structural differences. Whereas coming to this RNA, we are having three types, rRNA, tRNA, mRNA. rRNA is ribosomal RNA, which is present in ribosomes, which is responsible for giving a platform, workbench for the protein synthesis. tRNA is going to be useful for transporting the amino acids to the protein synthetic site. Whereas mRNA is very important for us, it's a genetic code providing one. Which amino acid has to come after which amino acid, that is going to be decided by your messenger RNA, that is mRNA. Now you will see structural differences between the RNA and DNA. Now structural peculiarities we are taking. DNA, I will call this one as polynucleotide. So this is polynucleotide, either DNA or RNA, both of them are going to be polynucleotide. So this DNA or RNA, if you are breaking it, what I am going to get? Nucleotide. I am going to get a nucleotide. If this nucleotide is going to get hydrolyzed, I am going to get nucleoside. This nucleoside is going to be get hydrolyzed. I am going to get nitrogen base plus sugar. So that is what is going to be making the difference between. Say for example, it's a DNA is going to be formed by deoxyribonucleotide. When you are going to hydrolyze this one, I am going to get deoxyribonucleotide. So this is going to be hydrolyzed. It is going to give deoxyribonucleoside. This is going to give deoxyribonucleoside. Now this is going to be hydrolyzed to give nitrogen base plus deoxyribose. Understand? This is what we can able to see the scenario in the structural peculiarities. The DNA is formed by deoxyribonucleotide. Deoxyribonucleotide is formed by deoxyribonucleoside. It is formed by deoxyribose and nitrogen base. That is what we are going to have. Whereas coming to RNA, it is also called as polynucleotide only. Now this is going to give ribonucleotide. I am going to get ribonucleotide. This is going to give ribonucleoside. So ribonucleoside is going to give you ribose sugar plus nitrogen base. This is what we can able to write for these compounds. Now the difference you can able to observe, ribo, deoxyribo. Now we will be taking from the bottom. Say for example, deoxyribose and nitrogen base are combined by a bond called glycosidic bond. We are going to have glycosidic bond. Just I am taking it in reverse. What bond we are going to have? Glycosidic bond. This glycosidic bond can also be called as N-glycosidic bond. We are going to call this one as N-glycosidic bond because what we are having in carbohydrate, that is going to have oxygen glycosidic bond, sharing of oxygen. Oxygen is going to form the bridge between your carbohydrate to carbohydrate. Here to carbohydrate the ribose and we are taking nitrogen base. The nitrogen is going to form the bridge. That's what is called as N-glycosidic bond. It's called as N-glycosidic bond is going to form nitrogen base combined with deoxyribose, so that you are going to get deoxyribonucleoside. Now, this deoxyribonucleoside is going to be combined with a phosphate. It's going to be combined with a phosphate. Phosphate, we can able to write it P with circle, or we can write with inorganic phosphate like Pi. Anything is going to be allowed. Now, these are going to be combined with deoxyribonucleoside by the linkage called 5' phosphoester bond. We are going to have 5' phosphoester bond, that is phosphoric acid is going to be an acid. A ribose sugar is going to have alcohol. That alcohol is going to combine with your acid, and we are going to call that one as acid plus alcohol is ester. That is going to be happens at 5th position, that is 5' position of ribose sugar. That's what is called as 5' phosphoester bond is going to form deoxyribonucleotide. Now, so many deoxyribonucleotides are going to be get polymerized or joined together by the bond called 3' 5' phosphodiester bond. So now you can able to very careful about the bonds what we are going to have between which one to which one. This is a question usually you are going to get it from the nucleic acid part. Now nitrogen base and a deoxyribose is going to combine by the bond called N-glycosidic bond. So the glycosidic bond is always going to combine the carbohydrate. Here nitrogen is forming the bridge, that's what is called as N-glycosidic bond. And now the deoxyribonucleoside, which is formed by the combination of nitrogen base and the sugar, is called as deoxyribonucleoside. This nucleoside is going to be combined with a phosphoric acid at 5th position by the bond called 5' phosphoester bond. So that what is going to be formed here is called as deoxyribonucleotide. Deoxyribonucleotide. It is side, it is tied. Then these deoxyribonucleotides are going to be combined with one another by the bond called 3' 5' phosphodiester bond, and which is going to form a polynucleotide chain that is representing the DNA. Bonds are same, but the wording is different. Ribose plus nitrogen base is going to form ribonucleoside by the bond called N-glycosidic bond, and then ribonucleoside is going to be combined with your phosphate by the bond called 5' phosphoester bond to form ribonucleotide. These are going to be get polymerized to give RNA. Again here bond is 3' 5' phosphodiester bond. In both the cases we are going to have the same thing by the bonding concern, but only what we are going to have the difference mainly is here I am going to have the ribose sugar, here I am going to have deoxyribose sugar. That is only the difference I am going to have in these two. That is the main difference. The remaining all the bonds and everything is same. Understand? So this is about structural difference. Now you will see the nitrogen bases and the ribose sugar structure and how they are going to form these nucleotides. Okay? So I hope everyone is getting clarity about these bonds. So we will see now how the nitrogen bases are going to be combining with your ribose sugar to form the nucleosides. So basically we are going to have two nitrogen bases. Purines pyrimidines. We are going to have purines and pyrimidines. Purines are the derivative of purine ring. This is purine ring. So the derivatives of this purine ring are called as purines, and this is called pyrimidine ring. Derivatives of this pyrimidine are called as pyrimidines. So what are purines we are having? A and G, adenine, guanine. So what is adenine? What is guanine? We can able to remember the chemical name of adenine. We can able to draw the structure easily. For knowing that one, we are supposed to know the numbering which is going to be present in your nitrogen bases, purine and pyrimidine. So the numbering in case of purine will start with here 1, 2, 3, 4, 5, 6. This is the numbering 1, 2, 3, 4, 5, 6, 7, 8, 9. So 1, 2, 3, 4, 5, 6, 7, 8, 9. This is the numbering in case of purine. In case of pyrimidine, the numbering will be 1, 2, 3, 4, 5, 6. This is the numbering in case of pyrimidine. This is the numbering in case of purine. Now I will be writing the chemical names of your purines and pyrimidines. 6-amino purine is called as adenine. What is adenine? 6-amino purine is called as adenine. So sixth position in purine, where it is? Here I am having, so that what I can write here? NH2. 6-amino purine. Now it is becoming adenine. Now one more I am going to have is this is one more purine ring. We are going to give the name for this one is 2-amino 6-hydroxy. 2-amino 6-hydroxy purine is called as guanine. So what is this one? 2-amino 6-hydroxy purine is called as guanine. Second position here OH. Here I am having, second position NH2, and sixth position I am having hydroxy. So 2-amino 6-hydroxy purine is nothing but guanine. Two purine bases over. One is going to be 6-amino purine, another one is 2-amino 6-hydroxy purine, adenine, guanine. That is the names, we know it already. So this is about purines. Now coming to the pyrimidines. The pyrimidines we are going to have three, that is uracil, cytosine and thymine. Now what we are going to have, first one is uracil, is going to be 2,4-dihydroxy pyrimidine. What it is? 2,4-dihydroxy pyrimidine. So now this is second position, this is fourth position. So 2,4-dihydroxy pyrimidine is nothing but uracil. Next one is thymine. just we are going to have one extra, that is 2,4-dihydroxy 5-methyl pyrimidine. 5-methyl pyrimidine is going to be thymine. So other than these two, we are going to add one methyl group here. Here OH, here OH, here CH3. So that's it, we are having the difference. What we discussed, nitrogen bases present in DNA are A, T, G, C, whereas in RNA is A, U, G, C. Uracil is having 2 hydroxyl groups, one methyl group is present in thymine, that is only the difference. Cytosine is common, so what is cytosine? So if you are taking a cytosine, just you can able to write 2, second position you are having hydroxy and 4th position you are having amino group. So 4 amino 2 hydroxy pyrimidine. So this one, the structure can be so that's it, we are having the structure of purines and pyrimidines. If we can remember the chemical names of those compounds, then it is very easy for us to get the name of a particular compound. So once again I am going to see here. Here it is 6 amino purine is adenine, 2 amino 6 hydroxy purine is guanine. These two are purine rings and the numbering is very important. And coming to the pyrimidine numbering is like this. We are going to have 2,4-dihydroxy is going to be uracil, 2,4-dihydroxy 5-methyl is thymine, whereas 4 amino 2-hydroxy pyrimidine is cytosine. These are the structures of your nitrogen bases we are going to have. And moreover, if you can see in our NCERT textbook, they are going to give, wherever I am writing OH, there they are going to write double bond O. So don't think that what I am writing here is wrong. Actually, they are going to show tautomerism. They are going to show tautomerism. When we are going to have OH and double bond, enol. Enol is going to show tautomerism. Say for example, for this one only I am writing the tautomeric structure. Now this double bond is shifted and this H will come here. So now here I am going to have double bond O and here I am having H. Similarly, here H is shifting here, I am going to have H and here I am having double bond O. Now what name I can able to give? 2,4-dioxopyrimidine. Earlier what I said, 2,4-dihydroxy pyrimidine is called as uracil. Now I can able to write 2,4-dioxopyrimidine. Why? Because it is existing in now the imide form. Actually they are called as cyclic imides. They are called as lactam, lactim. One is lactam, another one is lactim. They can able to show tautomerism. That's why wherever the OH is there, if you want to write double bond O, take out that adjacent double bond and keep double bond O, then it is going to be the structure. Whatever you are writing, either hydroxy or keto. Keto form is more stable, that's why they are writing usually here. But for us, all triple bonds are same. Then if you remember like hydroxy, the structure writing is easy. That's why I am always showing like all double bonds are constant. Here also the same thing, double bond O, NH you can write. But the student is going to get confusion. This is different structure, this is different structure. That's why I am keeping always a purine ring constant. That's why I am writing OH. For your idea, if you want to write keto form, this OH, H will come here and here double bond O I can write. Here also double bond O I can write. Here double bond O I can write. So that is going to be called as keto form. which is going to be shown by your pyrimidines and purines when they are going to be having some substituent on them, that is called as lactam tautomerism. So those things are not required for us. Just for your idea, I am saying their OH writing or double bond O writing, anything is same only. So these are all your purine bases and pyrimidine bases. How they are going to form the bond with your ribose sugar, we will see now. So what sugars we are going to have? Ribose and deoxyribose. Yes, I am writing sugar what ribose it is? Beta D ribose. OH, H, H, OH, H, OH. Here I am having HCH2OH. This is what I am having now. And here I has to say one more bonding called N1, N9. In case of purines, we will have N9 glycosidic bond. That is, 9th position hydrogen is going to be forming the bond between these two. So now what I am going to get here? So now where it is get binded? The 9th position is going to be get binded. Always it is going to be at first position of your ribose only, but the 9th position of purine is going to have, always purines will have N9 glycosidic bond. Purines will show N9 glycosidic bond. If you are taking pyrimidine, we are going to have this nitrogen is going to be existing in, I said already, it's going to be existing in keto form. Now we are having hydrogen here. Now this H and this OH will be going out. See here I am not writing OH here. Second position I am not having oxygen. Here it is not having oxygen, that's why it is going to be 2' deoxy beta D ribose. 2' deoxy beta D ribose. As you are having thymine, it should be present in DNA. So that's why you are going to have deoxyribose I am writing. So now what glycosidic bond is on? N1 glycosidic bond. Why? Because the pyrimidine's first position is going to be forming the bond with your ribose sugar. That's what is called as N1 glycosidic bond. Whenever you are going to have the glycosidic bonds, what you are supposed to remember it? If it is purine, we are going to have N9 glycosidic bond. If it is pyrimidine, we are going to have N1 glycosidic bond. It is fixed. Commonly I given N glycosidic bond when I am discussing that entire structure. But individually, whenever you are writing the structures, we are supposed to know the specifically where the number it is going to be get attached. N1 glycosidic bond is shown by pyrimidines with ribose sugars. N9 glycosidic bond is shown with purine and ribose sugar. As I wrote here deoxyribose because the difference we are going to have, thymine is going to show DNA, whereas uracil is going to be present in RNA. That's why for thymine I wrote deoxyribose. So that's why we are going to write. Now what we can call this one as? Nucleoside. That's what I said, no? Nitrogen base and the ribose sugar is going to be giving you nucleoside. This is also nitrogen base and your ribose sugar is going to be called as nucleoside. Now this nucleoside is going to form nucleotide when it is going to be, when it is going to form a reaction with your acid. I said no, inorganic phosphate can be shown with Pi or P with circle. It's indicating inorganic phosphate. Now it's an alcoholic group present at 5' position. In order to differentiate numbering here and here, 1, 2, 3, 4, 5, 6. And here we are giving like 1', 2', 3', 4', 5'. Just to differentiate numbering in sugar and the pyrimidine, we are giving primes. That's it, we are not having anything else. Now 5' position, CH2OH alcohol is going to react with a acid, phosphoric acid. This is going to be called as a water molecule is going to be comes out, so that the linkage which is formed between acid and ester is called as ester bond. And now it's a phosphoric acid, that's what it is called as phosphoester bond. It's not usually the carboxylic acid and alcohol is going to form ester, but here phosphoric acid and alcohol is going to form the ester. It's called as phosphoester bond. So because of this phosphoester bond, what formed here is called as nucleotide. It is called as nucleotide or nucleotide monophosphate. If you are going to add up one more phosphate, it's called as nucleotide diphosphate. One more is called as nucleotide triphosphate. Like this, we are going to give the nucleotide formation. Now we'll see how a nucleotide is going to be forming the bond with one more nucleotide. See, this is the simplified version of your nucleotide I am going to write here. It's a ribose sugar. It's going to have adenine. That means it is going to be, what it is? Nucleoside. Now this nucleoside is going to have one phosphoric acid. Then this is going to be called as nucleoside monophosphate. Now this is going to have one more phosphoric acid. Then it is called as nucleoside diphosphate. Then we are having one more. It's called as nucleotide triphosphate. NTPs. NTP, nucleotide triphosphate, which are going to be present adenine, ATP, adenosine monophosphate, AMP, adenosine diphosphate, adenosine triphosphate. So this is nothing but adenosine triphosphate. I think everyone knows about this one. It's an energy currency of cell, which is shortcut called as ATP. ATP is nothing but adenosine triphosphate. Adenosine triphosphate. Now, when we are going to take this adenosine monophosphate, diphosphate, triphosphate, what are the bonds we are going to have? That's what I want to discuss. This is called N9 glycosidic bond. Already I have shown. Just for your clarity, I am writing it again. This is called N9 glycosidic bond is present between the ribose sugar and adenine. Now CH2OH and phosphoric acid. Now this bond is going to be called as 5' phosphoester bond. This is called 5' phosphoester bond, which is present between alcoholic group of your ribose sugar and the phosphoric acid is called as 5' phosphoester. Now phosphoric acid is going to be joined to another phosphoric acid. What bond we can call this one? It's an acid, it's an acid. Now the two acids are going to lose the water, which is going to be called as N-hydride bond. This is called as N-hydride, and this is also N-hydride. We are going to get commonly the question, what number of bonds present in a ATP molecule? We are going to have N9 glycosidic bond, we are having 5' phosphoester bond, we are having a anhydride bond. These are the bonds which are present in one nucleoside triphosphate when you are taking. It's a best example, ATP, adenosine triphosphate, which is going to have all these bonds. Okay, now we will see how they are going to form a bond between one nucleotide to another nucleotide. That is how we can able to call that one as 3' 5' phosphodiester bond. Is it clear for everyone? I hope the bonds which are present in your nucleic acid, nucleotide, nucleoside are clear, isn't it? Yeah, we'll go to the next one, how they are going to form the bond. So now when we are going to take the two nucleotides, now what is going to be happen here? The nucleotides are going to be having phosphoric acid at 5th' position and your alcohol is getting at 3' position. Now you can give the numbering here 1', 2', 3', 4', 5'. Now this is going to be 5' for this one, 4', 3', 2', 1'. Now 5' position of one nucleotide is going to be having the phosphoric acid, 3' we are going to have OH, and now this OH and this OH will goes out as water and they are going to form a bond. Now how this bond is going to be formed here? 3', 5', phospho, here one ester bond, here one ester bond. Why? Because phosphoric acid with your alcohol, it's formed one ester bond, here also one ester bond. That's why it is called as 3', 5' phosphodiester bond, which is going to be forming, and when you are going to take, it will be continued like this. If we are taking like this, we are going to get the bond formation. That is how I can able to get the phosphodiester bond. If I want to write this entire thing, nitrogen base, HOH, here H, O, P, double bond O, and CH2, base, H, OH, H, here O, P double bond O, OH, O, CH2, H, base, HOH, HOH. So if you are looking at like this, it's going to have 5' position, I am going to have phosphate is free, 3' position I am having free hydroxyl group. So that's why whenever you are going to say the direction of nucleotide strand, we are going to call as 3', 5'. Excuse me. What you are going to call this one? 3', 5' position we are going to call, because the 3' is going to have one OH group free, whereas 5' we are going to have phosphate group free. That's why it is going to be said to be 3' to 5' or 5' to 3'. Whenever you are going to say the DNA strands, whenever one is going to be 3' to 5', another one is going to be 5' to 3', that's what is called as anti-parallel winding. The same thing we can able to say it here also, how it is going to be 5' position, one phosphate group is free, 3' position, one OH group is free. That is how we are going to have. So when DNA is going to be having, it is going to have two strands. So there we are not having this OH, and we are going to have base, and base will come side by side. Then they are going to form a DNA double strand. DNA double strand is going to be forming. So that's why the backbone for your DNA strand or RNA strand is going to be 5', 3' phosphodiester bond or 3' 5' phosphodiester bond, we can say it. Is it clear? The bonding what we are going to have in DNA. I hope everyone can able to say it now. Is it clear? Just I am repeating it. The nitrogen base and your ribose or deoxyribose will have the bond called N1 glycosidic bond in case of pyrimidine, N9 glycosidic bond in case of purine. Now the formed nucleoside is going to be forming the bond with phosphate. That is going to be called as nucleotide by the bond called 5' phosphoester bond. The formed nucleotides are going to be forming the polymerization, self-linking, that is going to be having the bond called 3' 5' phosphodiester bond. That is how we can able to get the backbone for your DNA or RNA strand is your 3' 5' phosphodiester bond. Understand? So this is what we are going to have, and only we are having one more left, like when we are going to give the name, the adenine is going to become what will be called as nucleoside, what its nucleotide called as. Just you will see that one, that will be the structure of your DNA and RNA. Adenine is going to be called as adenosine. If you are adding one phosphate, AMP, ADP, ATP. Already I said how we are going to get ADP and ATP. Just this is one adenosine. How we are going to get this one? Ribose is added. That is nucleoside, and this one is nucleotide is added by phosphate addition. This is also called as adenylate acid. This is called as adenylate acid or adenylate acid. This is going to be ribose sugar added. We are going to call them as adenosine. Same if you are taking guanine, we are going to call it as guanosine. Guanosine is going to become GMP, guanylinic acid. Whereas if you are taking cytosine, this is called as cytidine, CMP, cytidylic acid. It's called as cytidylic acid. Next one, thymine, thymidine, TMP, thymidilic acid. Next one, uracil, uridine, UMP, uridylic acid. These are the questions. What way we are able to get is, when we are going to take uracil, what it is going to be? It's a base, pyrimidine. If you are taking uridine, what it is? Uridine is a nucleoside which is formed by the ribose and this one. If you are using deoxyribose, it is deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymidine, deoxyuridine. Here it will become DAMP, DGMP, DCMP, DTMP, DUMP. We will add some D, small d. That's it. We are going to have the difference between the adenine, guanine, cytosine, thymine, uracil will become adenosine, guanosine, cytidine, thymidine, uridine. They will become adenylic acid, guanylic acid, cytidylic acid, thymidilic acid, uridylic acid. This is what we can able to understand. What we are able to call them when they are going to become your nucleosides or nucleotides. Understand? Now, what are the other characteristics of your DNA and RNA? I think everyone knows about them. So, DNA is going to have a double helical structure. How that helical structure will be? This is how the DNA will be present. Now, this double bonded structure or double stranded structure is going to be stabilized by a hydrogen bond between the nitrogen bases which are present. I think everyone knows about this one. How these hydrogen bonds are going to be formed? That is, adenine pairs with 2 hydrogen bonds by thymine, guanine pairs with cytosine by 3 hydrogen bonds. So, adenine is going to form 2 hydrogen bonds and this is going to be 3 hydrogen bonds. You are writing like this also, but always hydrogen bond is showing with a dotted line. I am writing like this. But you are supposed to have an idea what bond is going to be present between one guanine to one cytosine. They are not covalent bonds, they are going to be hydrogen bonds, they are going to be forming. Now, whenever a DNA is going to have more GC content, obviously that is going to be more stable because of more number of hydrogen bonds. With GC content, with AT content, which is more stable, with GC content is going to be more stable than with one which is having more AT content. And moreover, always adenine pairs with thymine. It's a pyrimidine, it's a purine. Guanine is always pairs with cytosine. It's a purine, it's a pyrimidine. Why not opposite? Why can't A with C, G with T? If you are going to get that one question, the A and C will not able to form the bonds. There is no hydrogen bond formation between adenine and cytosine. That's why you will never get that one. But G and T, if you are taking, it can able to form one hydrogen bond. G and T can able to form one hydrogen bond. That is more than we are having 2 hydrogen bonds here, 3 hydrogen bonds here. That's why always it is going to go for more hydrogen bonding capacity is there between GC and AT. That is only possible. As we are having purine always pairs with thymine, guanine always pairs with cytosine, we can able to say A plus T, number of purines we are going to have is equal to number of pyrimidines we are having in DNA. Chargaff's rule, A plus T is equal to J plus C. So that is what we are going to have. Purines and pyrimidines are going to have same when we are going to take a DNA. But for an RNA, this is not going to be same. Why? Because they are all not double stranded. They are going to be single stranded. Okay. So DNA is going to have one helices is going to be having the length of 36 angstrom units. Between 2 bases, we are going to have 3.6 angstrom units. 10 base pairs on an approximate one helices is going to have 10 into 3.6, 36 angstroms, and this diameter is going to be 20 angstrom units. These are all the things we are going to study in your biology. That's why I am not considering most of them. Just anyway you learn in your bio. So it is 36, it is 3.6, the diameter is 20 angstroms when you are taking a double helical structure of DNA. AT by GC is equal to 1 in case of, more than 1 in case of human being and 0.93 in case of E. coli. So that is what so many times they are asking the question, what will be the AT by GC content? In human beings it is approximately 1.52. In E. coli it is going to be 0.93 AT by GC ratio. These are the points we have to remember regarding the DNA. Coming to the functionality, I already explained, DNA is going to show the functionality like autocatalytic, that is going to have replication, and it is also showing transcription, and the RNA which is formed is nothing but blueprint of DNA that is going to give you the protein. This entire process is called as central dogma. Central dogma. Whereas some virus which is going to have the chance of making mRNA to DNA, which is called as reverse transcription, then that time it is going to be called as central dogma reverse. Central dogma reverse we are going to call that. So this is about your nucleic acids what you are going to have. Then we are having a small point regarding DNA melting. melting temperature of DNA. So what is this one? When the two strands of DNA is getting separated, the temperature is called as melting temperature. When we are going to cool that one, this is going to be get renatured, that is going to be called as annealing, and the DNA is going to be get lysed because of the hydrogen bonds are going to be get broken, that is called as melting temperature. That is called as melting temperature. That is what we are supposed to have an idea about DNA and RNA. And just I am going to give the RNA functionality is already said. What is the structure of tRNA? That is the most of the times we are getting. tRNA is going to have clover leaf structure. We are going to call this one as clover leaf structure. So the clover leaf structure is going to have some loops, and all those things are not important for us. Just remember, clover leaf structure will be there for tRNA, which is 3D. Whereas RNA, mRNA is going to have helical structure, single helical structure, and rRNA is going to be present in a linear form. rRNA is going to be present in linear form, mRNA is helical structure, tRNA is going to have clover leaf structure. That's what we can able to say it in case of RNA. RNA is going to show protein synthesis. The genetic code you learned, I think, in your bio thing. So the genetic code is going to give the 3-letter code. It's a universal code which is going to have some ambiguity is there. Wobble hypothesis is going to be explaining that one. A single one is going to be starting more than one. And whereas initiation codon is always going to be AUG. Some codons are not going to make the protein synthesis. They are called as nonsense codons, and which are going to have the remaining 61. Total 64 codons are there. Out of 64, 3 are nonsense codons. Remaining 61 can able to synthesize the amino acids in your protein synthesis. I hope everyone can able to understand this DNA and RNA, and only one thing we left with hormones. So what are hormones? Hormones are nothing but they are endocrinal secretions. These things we can able to see in tomorrow's class, then we can followed by the next chapter. So this is only the small part left in this biomolecules, that is hormones. We will see tomorrow. And this is about your DNA and RNA. What bonds I explained here, they are very important. Everyone should by heart them. Whenever the question comes from the nucleic acid, obviously that is from your bonding only. And one more application of DNA we are going to have, the DNA fingerprinting and the hormones. These two concepts we will be explaining tomorrow. So everyone should by heart what the bonds they are going to have between them, and the ribose and deoxyribose structures we are supposed to remember it, and the pyrimidines and purines, that they are going to ask usually which of the following is adenine, which of the following is guanine, which of the following cytosine. These are all the things are important for us. And if they given A plus T ratio, G plus C ratio, they are going to ask us AT by GC, just what they given, just we have to divide them, then we are going to get the answer. Okay, is it clear? And what are the doubts you are having, you can ask me during the interaction time. Thank you.